Hello friends, I am Dr. DP Bora. I welcome you to my YouTube channel, The Microbiology Digest. Today also we will be discussing another topic of immunology, that is the cells of the immune system. So in our last discussions we have covered the various organs that constitutes the immune system. Today we will be discussing the cells, different types of cells which are responsible for inducing the immune response against foreign antigens. So already we have discussed many times that immune system it comprises of various organs and tissues as well as various cells, and this is a highly regulated system which protects us from the infection by pathogenic microorganisms. So what are the cells that are responsible for generating the immune response? So to name these cells, mostly the lymphocytes, then macrophages, monocytes, dendritic cells, granulocytes. So these are some of the examples of cells which take part in the generation of the immune response. Now all these cells actually they arise from the pluripotent hematopoietic stem cell. They arise from a single cell which is called as hematopoietic stem cell, and these stem cells they are pluripotent. Now these cells actually they are present normally as a circulating cells in the blood as well as in the lymph, and almost most of the tissues they will be present, except these cells are not present in the central nervous system. Now the process by which these cells are formed from the stem cell, this process is called as hematopoiesis. This is the formation of cells. So all these blood cells actually they arise from a cell, from a single cell, that is called as hematopoietic stem cell. These stem cells they are pluripotent, means they can give rise to, or they can differentiate into many different cells. They can differentiate into many different cells, and they can give rise to many different cells. And every functionally specialized mature cells they actually arise from a hematopoietic stem cell only. So hematopoietic stem cells, as we have already discussed, they are pluripotent because they can differentiate into different cell types, and they are also capable of self-renewal. Self-renewal capacity is there, and they can maintain their population by cell division. These are some of the properties of the pluripotent stem cells. Now, early in the hematopoiesis, the process, these stem cells, pluripotent stem cell, they will be dedicated, or they will be differentiated to one of the two lineages. There will be primarily two lineages, or two ways by which these stem cells will be differentiated. So one lineage will be the lymphoid progenitor cell or lymphoid lineage cells, and the second type is the myeloid progenitor or myeloid lineages. So these progenitor cells, once these cells are differentiated, the hematopoietic stem cell, when it differentiates into either lymphoid progenitor or myeloid progenitor, they lost the capacity of self-renewal, and now they are committed to a particular lineage. Either they will be going for the lymphoid lineage cells, or they will be going for the myeloid lineage cells. At this stage, they lost the self-renewal capacity. Means they cannot go back to again stem cell. So lymphoid progenitor cells, what are the lymphoid progenitor cells? They give rise to the B cells, T cells, as well as the natural killer cells. Then what are the myeloid progenitor cells? So they are mostly the the red blood cells, erythrocyte, and other blood cells, white cells, neutrophils, eosinophils, basophils, they all generate from the myeloid progenitor cell. And this hematopoiesis, the entire process, it is a highly regulated process. It has to be controlled at a in a genetic level. So this is the diagrammatic illustration how a hematopoietic stem cell, it gives rise to two different cell lineages. One is lymphoid lineage, another one is myeloid lineage. And lymphoid lineage, it will give rise to the cells like T cells, B cells, and natural killer cells. And the myeloid lineage will give rise to the erythrocytes or other blood cells, which are the component of this myeloid lineage. Now, as I say, this hematopoiesis must be regulated. There are different mechanisms by which this hematopoiesis is regulated. So why it is required? Hematopoiesis is a continuous process. It is every time cells are being produced. But if it is not controlled or if it is not regulated, so more and more number of cells will be produced. But that is not required. So that is why there are certain mechanisms which controls the entire hematopoiesis process. So there are some lifespans. Suppose average erythrocyte, it has a lifespan of 120 days, and other blood cells also their lifespan is different. So this regulation, it is achieved by various mechanisms. For example, they will be controlling the levels of various cytokines, and they will be controlling or they will be regulating the activity of these cytokines, or these gene expressions will be controlled, and the controlled induction of a process which is called as programmed cell death. So where these cells will be periodically removed from the system. So these are mechanisms by which hematopoiesis is controlled by the, or it is highly uh, controlled through a regulated mechanism. Now here one point is to be understand what is programmed cell death, or it is also otherwise called as apoptosis. So this is an induced programmed cell death. Death is an induced and ordered process. So what happens here? These cells, they actively participates in bringing its own demise. Means it is the death of our own cells by a regulated mechanism, through a regulated mechanism. So it is called as programmed cell death. So this is, as we have already discussed, this is required, this is essential for regulation of the hematopoiesis. So these cells which takes parts in the programmed cell death, they will exhibit some of the changes in the structure. So distinctive morphological changes will be expressed by these cells, and these are collectively called as apoptosis. So what are these changes? There will be pronounced decrease in the cell volume. There will be modification of the cytoskeleton. There may be membrane blebbing. The cell membrane, there will be blebbing. Then there will be condensation of the chromatin. Then there may be degradation of the cellular DNA into various small fragments. Okay, so once these changes are exhibited by the cells, participating cells, so these cells, they will be, these changes will be expressed in the membrane, and finally these cells will be engulfed by the macrophages. So macrophages, they can quickly, once these changes are expressed by the cells, participating cells, despite macrophages, they will come into play and they will quickly induce phagocytosis of those cells. So here in one point is very essential to understand what is apoptosis and what is necrosis. Okay, so necrosis is also a process where there will be cell death, and This apoptosis is also a process where there will be cell death, but there is difference. So simple difference you can understand that apoptosis, it is a physiological process. So on the other hand, necrosis, it is a pathological process. Okay, so there are many more other differences are there. You can find out these differences so that you can understand the different processes involved in the hematopoiesis. Now, first type of cell, coming to the cell again, which takes parts in the, or what are the cells of the immune system. So first type of cell or major cell population, they are the lymphocytes. So lymphocytes, these are the principal or central cells of the immune system. They are responsible for adaptive immunity as well as other immunological attributes of the immune system. For example, diversity, specificity, memory, all these these attributes are due to the these lymphocytes. So lymphocytes, they constitute around 20 to 40% of the white blood cells and 99% of the cells in the lymph. Means majority of the cell population in blood as well as in lymph, they are the lymphocytes. So lymphocytes, there may be three different populations of lymphocytes. First population is the B cells, second is the T cells, and the third one is the natural killer cells. They are also called as NK cells. So these B cells and T cells, so when they are not interacted so far with the antigen, so they are called as unprimed cells or naive cells. Okay, and they generally they are in a Z naught phase of the cell cycle. Okay, so once they interact with the antigen, these cells actually they enter the cell cycle and they enlarge in their size about 15 millimeter in diameter, and these cells are called as lymphoblasts. Okay, these lymphoblasts actually eventually they differentiate into different uh, cells or different other cell types, which will later either it will be differentiated as plasma cells, which are the effector cells of the B cell lineage, and the cytotoxic T cell or the helper T cells, which are the effector cells of the T cell lineage. Okay, so this is the sequence of the events which takes place in the development of the lymphocytes. Now here another term you will encounter frequently that is called as CD, or it stands for cluster of differentiation. So each and every cell, different cell lineages of the immune system or different stages of the lymphocytes, they can be distinguished by their expression of some of the membrane molecules, and which can be recognized by particular monoclonal antibodies. So those all the monoclonal antibodies that react with a particular membrane molecule, they are grouped together as a cluster of differentiation, or it is called as CD. So there are various there's a very long list of CD markers identified so far. So I have listed out here some of the very common CD markers which are present in different cells of the immune system. Now, as we have discussed, the lymphocytes, they have three different uh, types. They can differentiate into three different types. One is B cell, another one is T cell, and third one is the natural killer cells. So first we will be discussing the B cell. So B lymphocytes, where from it is getting the name? Because these cells, they develops and they get differentiated, and uh, maturation occurs in the bursa of Fabricius in case of birds and in the bone marrow in case of mammals. So from the bone marrow or bursa, these cells have been named as B lymphocytes. B for either bursa or bone marrow. So B lymphocytes are the cells which uh, differentiates and matures in the bursa in case of birds and bone marrow in case of other mammals. So these cells, B cells, are the only cells which are capable of producing the antibodies. Then these mature B cells, they expresses the membrane-bound immunoglobulin in their they displays the membrane-bound immunoglobulin, so which will be serving as the receptor for the binding of the antigen. Okay, so this antigen, when it will be interacted with the membrane-bound antibody, these cells, B cells, they 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 gets activated and they will start differentiation of these cells, and this differentiation will lead to development of either antibody producing molecules or cells or the memory cells. Okay, next type of lymphocyte is the T lymphocyte. So T lymphocytes, they are getting their name from the site of their maturation, that is the thymus. So T from thymus. So like B cells, these cells also they express membrane-bound molecules, but here this is called as TCR or T cell receptors. These are the membrane-bound molecules which are expressed in the T cell. So again, T cell, they have two different subpopulations. One subpopulation, it is called as helper T cell, or it is called as TH cell, means T helper cell. So these cells actually they expresses the CD4 molecule, and they recognize the antigens when they are bound to class 2 MHC molecule. Okay, they are also called as CD4 plus T cells. So if we say CD4 T cells, means we should understand that these are helper T cells. Okay, so similarly, the other subpopulation of T cell is the cytotoxic T cells. So these cells, they expresses the CD8 marker or CD8 molecule, and they recognize the antigen when they are attached to the class 1 MHC molecule. Okay, so similarly, the CD, uh, cytotoxic T cells are also called as CD8 positive T cells. Means CD8 positive T cells, it indicates the cytotoxic T cells. Okay, now T cell, T helper to cytotoxic T cell, this is a we can uh, determine the ratio, but normal human being, the in human normal human peripheral blood, this ratio is generally, it has been estimated as a 2:1 ratio. Okay, now in response to the antigenic stimulation, these helper T cells, so they will be producing some of the protein hormones which are called as cytokines. Means cytokines are produced by helper T cells, and cytotoxic T cells, they are responsible for the destroying the foreign antigen molecules. Okay. Now third type of B lymphocyte, lymphocyte is the natural killer cells or NK cells. So these cells, first they were described in 1976, and they generally do not express any classical lymphocyte markers. Okay, and these cells, they express the CD16, which is a membrane receptor for carboxyl terminal of the immunoglobulin molecule. So because of this, they can take part, or they can exert their action through a specific uh, method called as antibody dependent cell mediated cytotoxicity. In short, it is called as ADCC. So natural killer cells, they exert their action through antibody dependent cell mediated cytotoxicity, and this is due to the CD16 molecule, which is expressed by the natural killer cells. Okay. Now, second type of cells, which are the which are cells of immune system. First type we have discussed about the lymphocytes, and second type of cells are mononuclear phagocytes. So, second population of cells of the immune system are mononuclear phagocytes. So, this is as I have said, or I have discussed, these are the second major cell population of the immune system, and these consist of the monocytes circulating in the blood and macrophages in the tissues. So what are the components of mononuclear phagocytes? They are monocytes when they will be circulating in the blood, and when they will be entering to various tissues, they are called as macrophages. Now, these macrophages, they are present in different tissues, and according to the tissues where they are present, they have been named differently. Suppose if these macrophages are present in the lung, they are called as alveolar macrophages. If they are present in the connective tissue, they are called as histiocytes. If they are present in liver, these cells are called as Kupffer cells. The cells which are present in the kidney, they are called as mesangial cells. If they are present in the brain, they are called as microglial cells. And if they are present in the bone, these macrophages are called as osteoclasts. Okay, these macrophages, they are distributed throughout the body, and they take active part in the elimination of the foreign antigens. So this is the diagrammatic illustration. Now, third type of cells of the immune system are granulocytic cells. So what are the granulocytic cells? So three different types: neutrophils, eosinophils, and basophils. So neutrophils, they are multi-lobed, they are having a multi-lobed nucleus, then granulated cytoplasm, and these cells are also called as polymorphonuclear leukocytes. They are phagocytic, and they constitute around 50 to 70% of the circulating white blood cells. Okay. Then second type is the eosinophil. They are also bilob. They are having a bilob nucleus, and they are phagocytic. They are also phagocytic, but they plays a crucial role against the parasitic organisms, or when these parasitic immune response against parasites are initiated. So eosinophils, they take or they serve a major function. Then third type is the basophil. They are having a lobed nucleus and heavily granulated. Okay, so here the neutrophils as well as eosinophils, they are phagocytic cells, whereas the basophils, they are not phagocytic cells. Now another cell population is the mast cells. These mast cells, actually, they are formed in the bone marrow during the hematopoiesis, and they are released into the blood as undifferentiated cells. So mast cells, also like other cells, they are generated during the hematopoiesis, and they are released into the blood as undifferentiated cells. Then they do not differentiate until they leave the blood and enter into the tissues. So mast cells can be found in different types of tissues like skin, connective tissue, different organs. There, these mast cells can be present, and these mast cells, actually, when it enters to the tissue, they take the name mast cells, and while they are in the circulation, they are the basophils. So these mast cells, along with the blood basophils, they are mostly important in case of the allergic conditions. Okay. The another type of cell, that is the dendritic cells. So dendritic cells, their name has been given because of their typical structure with long membrane extensions, which resembles the dendrites of the nerve cells. So because of that, these cells have been named as dendritic cells. So they are serving the purpose of antigen presentation. They are a very efficient antigen presenting cells. Dendritic cells, they are they are antigen presenting cells. So different four major group of dendritic cells have been recognized. That is the Langerhans dendritic cell, interstitial dendritic cell, monocyte derived dendritic cell, and the plasmacytoid derived dendritic cell. These four are the major groups of dendritic cells which have been identified. Then similarly, one more type of cell, that is called as follicular dendritic cells. Although the name dendritic cell has been attached here, these are not responsible for the antigen presentation. In fact, these cells they are actually involved in the B cell maturation, and they are mostly found in the lymph nodes in a particular location called lymphoid follicle. So these cells are present in the lymphoid follicles mostly. Lymphoid follicles, as we have already discussed, they are very rich in B cells. So these cells, they are present in the lymphoid follicles, and they do not serve as the antigen presenting cells. Please remember, follicular dendritic cells, although their name is similar with other dendritic cells, they are not uh, performing their function as a antigen presenting cell. Okay, so they also do not express the class 2 MHC molecule, and because of which they do not function as antigen presenting cell. So this is very interesting because the name is follicular dendritic cell, and we have already discussed that dendritic cells are efficient antigen presenting cells. So many a times we may get confused uh, by the name, but please remember, this is follicular dendritic cell is not an antigen presenting cell, and it also does not express the MHC class 2 molecule. Okay, so this is all about in a nutshell about the various cells which constitute the immune system, and which uh, functions in a highly regulated manner to mount or to induce an immune response against the foreign antigen. So I hope this information will be helpful, and if you find this is helpful, please subscribe to my channel. And with this, thank you very much. Thank you.